Genetic engineering is the modification of one organism's genome by the addition of another organism's DNA using biotechnology. It has many practical applications, including use in agriculture and farming, and in human genetic research and disease prevention. This presentation will include modern processes used in genetic engineering, examples of genetically modified organisms, and the ethics of genetic modification, including that of humans. Genetic engineering through biotechnology is a relatively new field. Before its introduction in the 1970s, humans bred animals and crops to increase the frequency of desired traits and remove undesirable ones. This sort of breeding, known as domestication, dates back to the time of the hunter-gatherers. The earliest known breeding of animals is with wolves, which brought along the appearance of the domesticated dog. The earliest evidence of this is in 14,000-year-old remains of what scientists believe to be an early domesticated dog, which now resembles what we now know as a sheepdog. Although these are the oldest remains, molecular divergence evidence suggests that humans had begun breeding wolves up to 100,000 years ago. Roughly 10,000 years ago, as humans started to make the transition from hunter-gatherer to farmer, crop breeding came into the forefront. Crops were bred for their favorable traits. This process occurred repeatedly over the next 10,000 years, giving us the crops we eat today. One of the pioneers in understanding the science behind breeding was German monk Gregor Mendel. His paper, published in 1866, Experiments on Plant Hybridization, cataloged his 8-year experiment dealing with heredity in pea plants. He coined the terms dominant and recessive genes and postulated the laws of inheritance, including segregation and independent assortment. Because of his groundbreaking work in genetics and heredity, he is known as the father of modern genetics. The field of genetics has come a long way since 1866. In 1972, Paul Berg devised a method for the first recombinant DNA using the monkey virus SV40 and the Lambda bacteriophage. His plan used restriction enzymes to cut out genes from the Lambda virus and implant them into the plasmid of the SV40 virus, using DNA ligase to seal the plasmid. Unfortunately, the danger of creating a genetically altered virus being able to spread through bacterial agent was enough to cause Berg to defer that part of the experiment to someone else. Despite this, Berg did create an effective method for recombinant DNA and was awarded the Nobel Prize in Chemistry in 1980. Berg's techniques did not go unused. In 1973, Herbert Boyer and Stanley Cohen created the first transgenic organism using Berg's methods for recombinant DNA. They used genes from two variations of antibiotic-resistant E. coli, one which was resistant to tetracycline and the other resistant to kanamycin. Using Berg's technique, they inserted the kanamycin resistance gene into the tetracycline-resistant E. coli and grew colonies of bacteria that were resistant to both antibiotics. In 1974, Rudolf Jaenisch created the first transgenic animal by adding the SV40 genome into the genome of a mouse embryo, then by implanting the embryo into a surrogate mother to allow it to develop. At first he was disappointed because he was unable to determine if the virus's genome had successfully added. A few months later, on the advice of Paul Berg, Jaenisch used a radioactive DNA probe to determine if the mouse's genome had been altered. This technique allowed him to see that he had indeed successfully added the SV40 virus into the genome of a mouse. In 2003, the first transgenic pet was introduced to consumers. The glowfish is a genetically modified zebrafish that contains the GFP gene, or green fluorescent protein. The gene is typically taken from marine animals and inserted into the genome of a zebrafish using recombinant DNA technology. This allows the fish to glow bright green when exposed to ultraviolet light. However, variations of the GFP gene are also used which produce different phenotypes, including yellow, red, and blue. The glowfish was originally designed to detect pollution in waterways, with fluorescing fish an indication of pollution. Due to their altered genome, glowfish cannot survive in the wild. They are an easy meal for any predators due to their high visibility. They also have reduced fertility and inefficient energy use because of the high energy cost of producing fluorescent proteins. Overall, glowfish are a novelty in the grand scheme of genetic engineering. Recombinant DNA technology is the entire basis for genetic engineering. It involves the insertion and incorporation of a foreign DNA sequence into the genome of another organism. There are three primary methods in which scientists do this. The first is transformation. Transformation starts by the selection of a DNA sequence of interest, which is generally called the donor. The donor is the foreign DNA that will be inserted into the vector. The vector is the carrier of the donor fragment, and most commonly takes the form of a bacterial plasmid. Importantly, expression factors must be present in the vector in order for the host cell to be able to transcribe and translate the protein specified by the donor sequence. These factors include the promoter, the ribosome binding site, and the terminator. The donor sequence is cut by a specific restriction enzyme, then ligated into the vector by DNA ligase. Once the recombinant DNA is created, it goes through the process of transformation, which is the insertion of the recombinant DNA into a host. Most common host is bacteria such as E. coli. The bacteria must be chemically treated in order to increase their DNA uptake from the environment, so when the recombinant DNA is added, they incorporate it into their genome. The bacteria are then placed in a growth medium to multiply, passing down their new recombinant DNA to their offspring every time they replicate. The second method of creating recombinant DNA is non-bacterial transformation. The process is very similar to transformation, except it does not use bacteria as a host cell. Instead, the recombinant DNA is inserted directly into the non-bacterial host by one of two common methods. The first is pronuclear microinjection, which is literally an injection of DNA into the host cell's nucleus using a glass micropipette. The second is by biolistic particle delivery system, also known as the gene gun. It fires small particles of gold or tungsten that have been coated with the recombinant DNA into the host cell. The third method of recombinant DNA is phage introduction or transfection. It is the equivalent of transformation except with the use of a virus rather than bacteria. The most commonly used viruses for this process are the lambda and M13 phages. Genetically modified organisms have many applications, especially agriculture and farming. In 1987, the Flavor Saver tomato was introduced to the consumer market. This tomato had a longer shelf life than ordinary tomatoes due to the lack of the enzyme polygalactoranase, or PG. This enzyme is known to break down pectin, which makes up the cell wall of plants. The breakdown caused the fruit to soften. The creators of the Flavor Saver had introduced into the tomato genome a reverse copy or antisense version of the gene that codes for PG. This antisense reduced the formation of PG, which allowed for much longer shelf life. The Flavor Saver tomato sold well until around 1998, where unwarranted customer concerns about safety, along with high production costs, declined the sale of the tomatoes drastically.
Much used in research, genetically modified organisms have many practical applications, particularly in the field of agriculture and food production. Industrial companies have perfected varieties of plants like maize, soya and cotton, which are resistant to parasites, fungi and diseases, or to certain harmful herbicides used in farming. Other GMOs are designed to improve the nutritional value of crops or to have beneficial health implications. For example, American researchers are working on a genetically modified banana, which acts as a vaccine against gastroenteritis. But GMOs are also a cause for concern. While there's no concrete proof they're harmful, environmentalists say more research needs to be carried out into their possible health risks.
Another genetically modified organism used in agriculture is the EnviroPig. The EnviroPig is a Yorkshire pig that has been modified to produce the enzyme phytase in its saliva. This modification was made to reduce phosphorus pollution brought on by raising pigs. Pigs normally require phosphorus supplementation because they cannot digest the phosphate present in their cereal grain food. The EnviroPig can digest this phosphate because phytase breaks down phosphorus in their food. The digestion of this phosphate causes less phosphate to be present in the pig's manure, reducing the risk of phosphorus pollution into the fresh water of surrounding areas. In order to add the phytase gene to pigs, the E. coli phytase gene was paired with the parotid secretory protein promoter sequence from a mouse to form a transgene construct. The construct was then inserted into the pig embryo through pronuclear microinjection, and the embryo was put into a surrogate mother. Through breeding, these pigs pass down their pollution-reducing trait to their offspring. Despite being one of the most promising fields in terms of genetic research and disease prevention, genetic engineering is very controversial. There are many people who oppose and support genetic engineering, with valid arguments being presented on both sides. Those with the pro-GMO stance maintain that increased crop yield and pest resistance will be able to help us combat world hunger or possibly decreased pollution, as with the EnviroPig. The anti-GMO side tends to believe that GMOs are harmful to humans, or more commonly that there has not been enough testing done on them to see if they are harmful. Another argument by anti-GMO groups is the environmental impact of genetically modified crops and animals. They are concerned that the accidental introduction of GMOs into the natural environment may disrupt ecosystems due to their unnatural characteristics or capabilities. One of the most convincing arguments by the anti-GMO stance is that we simply don't have enough knowledge of genetics to safely create transgenic organisms. They believe that there is a risk of unpredictable and possibly dangerous side effects from crossing unrelated organisms. One ethical issue that may present itself in the near future is the genetic modification of human beings. There are many benefits of successful human genome manipulation, including the possible prevention of genetically linked diseases such as cancer, sickle cell anemia, Huntington's, Down syndrome, and muscular dystrophy. There are also many risks and unforeseen side effects that come with this due to our limited understanding of genetics at this point. The primary goal of human genetic modification will be to improve the lives of people who will be dealt a bad hand in terms of their genetics. But in the light of this technology, many other options may become available to parents, including modification for aesthetic purposes or for superior physical capabilities and intellect. This raises many questions that we cannot answer currently. Where will we draw the line between modifications for disease prevention and modifications for more shallow purposes? What would be the repercussions if we tried to create a superior human being or a super soldier? Only the future holds the answer to questions like these. The introduction of this technology may also create social issues that we may not be prepared for, as discussed by Arthur Kaplan.
And there could come a day, probably an interesting argument against enhancement, where somebody would say, going back to an earlier question, well, why would I want to pay for your child with muscular dystrophy? You chose not to use improvement and enhancement. That's your problem. You chose that. You didn't do the genetic repair. You didn't do the therapies that could have prevented these problems.
The future of genetic engineering poses many questions that we just don't know the answer to, and it is something that we may have to deal with when the time comes.